Translation, also known as protein synthesis, is the mechanism in which the nucleotide sequence of information in messenger RNA is used to produce the amino acid sequence of a protein. We are now in the home stretch of our run through molecular biology, ready to begin a discussion of translation, also known as protein synthesis. Again, we need to remind ourselves that in this process, messenger RNA is not chemically converted into protein. Rather, the messenger RNA carries a nucleotide sequence information that can be translated into a sequence of amino acids in the final protein product. There are a number of features of the genetic code which we should consider. A sequence of 3 bases in messenger RNA carries the code for one amino acid in the final protein product. Alternately, one could also say that in the process of protein synthesis, a triplet of nucleotides called a codon in the messenger RNA is translated into one amino acid in the protein. The genetic code is non-overlapping, which means that a given stretch of messenger RNA has only one reading frame and only codes for one protein. The messenger RNA is generally translated 3 bases at a time from a fixed point to yield a single defined protein product. The genetic code is comma-less. That means that codons of 3 bases are read continuously with no spacer bases in between them. The first 3 bases are read, then the next 3, and the next 3, and so on and so on. The code is universal, meaning that all organisms use essentially the same code. The same codons specify the identical amino acids in the most ancient single-celled organisms and also in humans. There are a few minor exceptions to this generalization. The code is degenerate, indicating that several different codons specify the same amino acid. Simple math suggests that the use of 4 different bases to form 3 base codons would give 64 different combinations. Because there are 64 possible ways to code for 20 amino acids, the degeneracy of the code is not too surprising. The code is read from the 5' to the 3' end of the messenger RNA. We have seen that both DNA and RNA are synthesized from 5' to 3'. The reading of the messenger RNA also occurs in that same 5' to 3' direction. Let's take a minute to review the triplet codons that specify various amino acids. There are 64 possible codons that can be generated, with 4 different bases combining in any order to form triplets. It turns out that 3 of these are nonsense or stop codons, which code for the termination of translation. That is, they do not code for any amino acid, and when the translation machinery reaches one of these 3 codons, translation ceases. You should know these stop codons, which are UAA, UGA, and UAG. You should also know the codon for methionine, which is AUG. That methionine is generally the first amino acid to be incorporated into a protein. Mercifully, we'll spare you from learning the rest of the code, unless you really want to. The way this matrix works is that the first letter, the 5' position of the triplet codon, is given on the left side of the box. The second letter of the codon is on top of the box, and the third letter, the 3' position, is on the right side. For all 3 letters of the triplet, the bases are arranged in the order UCAG. Several kinds of RNA play key roles in translation. Messenger RNA is the template for protein synthesis. A messenger RNA molecule may code for individual genes in bacteria, but more often the messenger RNA codes for a cluster of related genes. A distinct messenger RNA is produced for each gene in higher eukaryotes. The messenger RNAs comprise a heterogeneous class of molecules whose size reflects the size of the proteins that they encode. In E. coli, the average length of a messenger RNA molecule is about 1.2 kilobases. Transfer RNA carries amino acids in an activated form to the ribosome for peptide bond formation. The recognition of the appropriate amino acid during translation involves a complementation between an anticodon triplet on the tRNA and a codon triplet on the messenger RNA template. There is at least one kind of tRNA for each of the 20 amino acids. The tRNAs are the smallest of the RNAs, with a length of about only 75 bases. Ribosomal RNA constitutes the major component of ribosomes. The ribosomal RNAs play both a catalytic and structural role in protein synthesis. In the E. coli ribosome, there are three kinds of ribosomal RNA called 23S, 16S, and 5S RNA. One molecule of each of these species of ribosomal RNA is present in each ribosome. Protein synthesis involves the translation of nucleotide sequences into amino acid sequences. Translation is mediated by the interplay of more than 100 macromolecules, including messenger RNA, ribosomal RNAs, transfer RNAs, amino acyl tRNA synthetases, and protein factors. Translation is an accurate process in which the incorporation of an incorrect amino acid occurs less than 10 to the minus fourth times. Transfer RNAs are the small adapters of about 75 nucleotides that link the triplet code in messenger RNA to an amino acid in the protein. Translation takes place on ribosomes, which are ribonucleoprotein particles, about 2/3 RNA and only 1/3 protein, consisting of large and small subunits. In E. coli, the 70S ribosome of 2,700 kilodaltons is made up of a small 30S and a large 50S subunit. The 30S subunit consists of a 16S ribosomal RNA and 21 different proteins. The 50S subunit consists of 23S and 5S ribosomal RNA and 34 different proteins. The messenger RNA is translated in the 5' to 3' direction, and proteins are synthesized in the amino to carboxyl terminal direction. The start signal for prokaryotic messenger RNA is AUG, or sometimes GUG, preceded by a purine-rich sequence that can base pair with the 16S ribosomal RNA. In prokaryotes, transcription and translation are closely coupled. Several ribosomes can simultaneously translate the same messenger RNA, forming a polysome. Protein synthesis takes place on ribosomes, which are ribonucleotide particles. Again, they're about 2/3 RNA, more RNA than protein, and only about 1/3 protein. They consist of one large and one small subunit. Prokaryotic organisms have a 70S ribosome with a total mass of 2,700 kilodaltons. In eukaryotes, there's an 80S ribosome with a total mass of 3,500 kilodaltons. This slide shows the substructure of the E. coli 70S ribosome. The smaller 30S subunit consists of a 16S ribosomal RNA component and about 21 different proteins. The 50S subunit consists of one 23S ribosomal RNA and one 5S ribosomal RNA and at least 31 different proteins. The large and small ribosomal subunits can exist independently and are brought together on the messenger RNA when protein synthesis occurs. Here's the complete sequence of the 16S ribosomal RNA component of the smaller 30S ribosomal subunit. It contains 1,542 nucleotides. The 16S ribosomal RNA consists of one continuous strand with one 5' end and one 3' end. This is single-stranded RNA, but you can see that significantly more than half of the bases in the RNA are hybridized to complementary bases in the same molecule. There are many complementary double-stranded stretches of RNA with a loop at the end. These are referred to as stem loop structures. This tendency to hybridize within the same chain is characteristic of many RNAs. A closer view of part of the 16S ribosomal RNA reveals that the complementation within the stems is not 100%. There appear to be a significant number of mismatches. Many of those mismatches consist of a GU combination, which would still be a purine-pyrimidine pair, just as in the correct GC or AU pairs. It seems as if the purine-purine pairing would be much more disruptive because of the accompanying steric problems. The structure of the 50S ribosome has been characterized by X-ray crystallography. The X-ray data suggests that there are various proteins around the periphery of the ribosome. In the center core are the 23S ribosomal RNA and the 5S ribosomal RNA molecules. It appears that the site of peptide bond synthesis lies within a central cavity that contains only the two ribosomal RNAs and no protein. Thus, peptide bond formation is most likely catalyzed by ribosomal RNA without the direct involvement of any catalytic protein. It's been speculated that primitive organisms may have used only RNA to catalyze protein synthesis, and that the ribosomal protein components were added later in evolutionary history. Data from extensive X-ray studies have been used to construct a model of the overall shape of the ribosomal assembly. The 50S subunit looks like a baseball glove. The 30S subunit resembles a legless ghost. The combined 70S ribosome suggests a modernistic attempt to portray the Madonna and Child. Transfer RNAs function as carriers of activated amino acids, and they contain anticodon regions that recognize distinct codons in the messenger RNA. There is at least one tRNA for each of the 20 amino acids. For many of the amino acids, there's more than one tRNA. Because every organism has at least 30 tRNAs, and some have as many as 50, and there are only 20 amino acids, it follows that some amino acids have 2 or possibly more tRNAs. The tRNAs are relatively small, about 70 to 80 nucleotides. They often contain distinctive rare bases, including methylated derivatives of A, U, G, and C, pseudouracil, and dihydrouracil. There are 3 stem loop structures in tRNAs. The loop designated DHU contains the modified base dihydrouridine. The anticodon loop contains the nucleotide triplet that recognizes a specific amino acid codon on the messenger RNA. Here's a generic tRNA. Each box in the structure represents a nucleotide. You can see the 3 stem loop structures that we noticed before in ribosomal RNA. The dotted lines on the stems show where complementation occurs between the bases. The overall shape of the tRNA is referred to as a cloverleaf or cruciform structure because of its cross-like appearance. The tRNA has 3 stem loop arms. The DHU loop contains a dihydrouridine residue. The anticodon loop has a nucleotide triplet, the anticodon, which recognizes a specific amino acid codon in the messenger RNA. This anticodon-codon recognition contributes to the amino acid specificity of a given tRNA in the translation process. The T-pseudouridine C loop is involved in binding to the ribosome. In addition to having the rare base of pseudouridine, this sequence also has a T, which is normally not found in RNA. There's also a small extra arm which varies in size depending on the exact number of nucleotides in the specific tRNA. At the top of the structure is a stem that has both ends of the tRNA. The 3' hydroxyl group on this stem ends in the sequence CCA, and it serves as the site of amino acid attachment. The structure shown on the right is dihydrouridine. The double bond found on the uridine ring has been reduced to dihydrouridine. Whereas the uridine ring is planar, the reduced ring of dihydrouridine is somewhat bent because it's no longer aromatic. This makes dihydrouridine less capable of hydrogen bonding with other bases. On the right in this slide we see pseudouridine. This derivative has the same basic ring system as uridine, but the point of attachment between the base and ribose is changed. Instead of a C-N bond, an N-glycosidic bond, we now have a C-C bond. The pseudouridine ring is still theoretically available for hydrogen bonding, but the specificity is changed. Whenever you see a Greek letter psi in an RNA, it specifies the occurrence of pseudouridine. Although transfer RNAs are often represented in a cloverleaf or cruciform shape, the actual folded structure of these molecules is more like an inverted L. On the upper right is the CCA end of the L, with the 3' end to which the amino acid becomes attached. On the end of the lower loop is the anticodon sequence that recognizes the messenger RNA codon. Somewhere in the middle of the transfer RNA, the T psi C loop and the DHU loop are folded near to each other. The process of protein synthesis occurs in 4 stages. First, the 20 different amino acid substrates undergo activation by the formation of covalent amino acyl tRNA derivatives. The important feature of this process is that the correct amino acid is charged onto the appropriate transfer RNA. Next, there's a specific and accurate mechanism for the initiation of protein synthesis. Generally, this occurs at the first AUG next to a purine-rich Shine-Dalgarno sequence in the messenger RNA. Elongation of the peptide chain is an energy-requiring process which occurs on and is catalyzed by ribosomes. The elongation process requires a messenger RNA template and various charged transfer RNAs. Termination of protein synthesis occurs when the translation machinery encounters the stop codon in the messenger RNA. Amino acyl transfer RNA synthetases read the genetic code. Each amino acid is activated and linked to a specific transfer RNA by an ATP-dependent enzyme called an amino acyl tRNA synthetase. The amino acyl tRNA synthetases link the carboxyl group of an amino acid to the 2' or 3' hydroxyl group of the adenosine unit of a CCA sequence at the 3' end of the tRNA. That's an ester linkage. There's at least one specific amino acyl tRNA synthetase and at least one specific tRNA for each amino acid. A synthetase uses both the functional groups and shape of its cognate amino acid to prevent the attachment of the incorrect amino acids to the tRNA. Some synthetases have a separate active site at which incorrectly linked amino acids are removed. A synthetase recognizes the anticodon, the acceptor stem, and sometimes other parts of its tRNA substrate. The covalent attachment of an amino acid to a transfer RNA is an energy-requiring process that's coupled to the hydrolysis of ATP to AMP and inorganic pyrophosphate. An amino acyl AMP serves as an activated intermediate in this process. The figure shows the 3 terminal nucleotide units at the end of a typical transfer RNA. Notice the nucleotide sequence is CCA. The amino acid is covalently linked to the 3' hydroxyl of a ribose by an ester bond. There are 2 evolutionarily distinct classes of synthetases, each recognizing 10 amino acids. The 2 classes recognize opposite faces of the transfer RNA molecules. The initiation of transcription occurs downstream from a purine-rich Shine-Dalgarno sequence on the messenger RNA. This sequence binds to a complementary region on the 16S component of the 30S ribosome. The first AUG codon downstream from the Shine-Dalgarno sequence is recognized by the tRNA carrying N-formylmethionine. Initiation also involves various proteins, the 50S ribosomal subunit, and it requires GTP hydrolysis. In prokaryotic organisms, N-formylmethionine is generally the first amino acid at the N-terminal end of proteins. In most prokaryotic genes, an AUG codon downstream from the Shine-Dalgarno sequence is recognized by a special tRNA, tRNA-F, carrying N-formylmethionine. Turns out that this AUG codon at the 5' end of the coding sequence is the only AUG in a gene that's read as N-formylmethionine. The other internal AUG codons are recognized by a different tRNA, tRNA-M, that carries and delivers the normal methionine residue. The biosynthesis of N-formylmethionyl tRNA involves an extra step. Methional tRNA is formed by a specific methional tRNA synthetase. Then in a second reaction, a 1-carbon formyl group is added to the alpha amino group of the methionine residue by a transformylase enzyme. The formyl group donor in this reaction is the coenzyme N-formyl tetrahydrofolate. The N-formyl tetrahydrofolate coenzyme is a derivative of the vitamin folic acid. This slide shows the complex process of ribosomal assembly that precedes the initiation of protein synthesis. In prokaryotes, a messenger RNA, the FMET tRNA, F, GTP, and a 30S ribosomal subunit all come together with the assistance of three initiation factor proteins, IF1, IF2, and IF3, to form a 30S initiation complex. The 30S ribosomal subunit binds specifically to the Shine-Dalgarno sequence, shown in red, near the 5' end of the messenger RNA. That orients the FMET tRNA on the first AUG codon, shown in green. At that point, IF3 has completed its task, and it diffuses away. Next, the 50S ribosomal subunit is bound, and IF1 and IF2 are released. The hydrolysis of GTP to GDP and inorganic phosphate also occurs in this phase. The complete 70S ribosome is now assembled, and the FMET tRNA is at the right place on the first AUG codon, and so everything is ready for the elongation phase of protein synthesis. This slide shows the start site of protein synthesis. You can see the Shine-Dalgarno sequence on the messenger RNA in its base pairing with a 7-base sequence UCC-UCCA near the 3' end of the 16S ribosomal RNA. Notice also the proximity of the Shine-Dalgarno sequence to the AUG code word for initiation. The importance of the Shine-Dalgarno sequence is that it results in initiation at the nearest AUG. That ensures that protein synthesis will proceed from the correct start codon and in the right reading frame. If initiation were to occur at some other AUG codon further downstream, it is likely that elongation would occur with the wrong reading frame, which would produce a meaningless truncated protein. Before we look at the process of peptide synthesis, review one more time the triplet codons that specify various amino acids. There are 64 possible codons that can be generated with 4 different bases combining in any order to form triplets. Remember, 3 of these, UAA, UGA, and UAG, are nonsense or stop codons, which code for the termination of translation. Also remember the codon for methionine is AUG. That methionine, or informal methionine, is generally the first amino acid to be incorporated into a protein. The process of elongation involves the addition of sequential amino acid residues to the growing peptide chain. The high accuracy of elongation is a function of the specific base pairing between the anticodon triplet on the amino acyl tRNAs and the codon units on the messenger RNA being read. The transfer of amino acid residues from tRNAs to the growing peptide chain is catalyzed by the ribozyme, that is, the catalytic RNA activity in the core region of the ribosome. Elongation involves the formation of peptide bonds. The ribosome has 3 sites for tRNA binding called the A amino acyl site, the P peptidyl site, and the E exit site. With a tRNA attached to the growing peptide chain in the P site, an amino acyl tRNA binds to the A site. A peptide bond is formed when the amino group of the amino acyl tRNA attacks the ester carbonyl group of the peptidyl tRNA. On peptide bond formation, the tRNAs and messenger RNAs must be translocated for the next cycle to begin. The deacylated tRNA moves to the E site and then leaves the ribosome, and the peptidyl tRNA moves from the A site into the P site. The codons of the messenger RNA are recognized by, that is, they form base pairs with, the anticodons of the amino acyl tRNAs. Elongation takes place on the ribosome and is catalyzed by a ribozyme or catalytic RNA enzyme. The 50S component of the ribosome has 3 binding sites for tRNA called the A site, amino acyl site, the P or peptidyl site, and the E exit site. On this slide, we're looking at the initial formation of the first dipeptide in a protein, starting with the FMET residue. The codons of the messenger RNA are recognized by, that is, they base pair with, the anticodons of the amino acyl tRNAs. For the first FMET, the 3' UAC anticodon sequence of the FMET tRNA would recognize the AUG codon of the messenger RNA. With an F-met tRNA attached to the P site on the ribosome, the next aminoacyl tRNA binds to the A site. That amino acid is delivered to the A site by elongation factor Tu, also called EFTU, in a GTP-dependent reaction. A peptide bond is formed when the amino group of the aminoacyl tRNA at the A site attacks the ester carbonyl group of the F-met tRNA at the P site. This reaction is catalyzed by the ribozyme activity of the 23S RNA of the large ribosomal subunit. The reaction is catalyzed solely by RNA. There's no protein found in this active site. After peptidyl bond formation, the tRNAs must be translocated for the next cycle to begin. The deacylated tRNA moves from the E site, and the peptidyl tRNA moves from the A site into the P site. Translocation is catalyzed by elongation factor G, EFG, in another GTP-dependent reaction. At this juncture, the F-met tRNA diffuses away from the E site. The F-met amino acid 1 and 2 dipeptide occupies the P site, and the system is ready for another round of elongation using the same bevy of RNA, proteins, and nucleotides. Let's take a minute to consider the energetics of peptide bond formation. It takes two GTPs going into GDP and PI to drive the elongation phase of protein synthesis. In addition, it takes one ATP going to AMP and two anorganic phosphates to charge each amino acid onto its tRNA. That means it requires the hydrolysis of 4 phosphoanhydride bonds to forge one peptide bond during protein synthesis. According to the Wobble hypothesis, some aminoacyl tRNAs recognize more than one codon because pairing of the third base of a codon is less crucial than that of the other two. The Wobble hypothesis was proposed by Francis Crick back in 1966. He suggested that during translation there's some leeway or wobble in the reading of the third base in a codon. The idea is that the 5' base on the anticodon triplet, which binds to the 3' base on the messenger RNA triplet, was not as spatially confined as the other two bases and could thus exhibit non-standard base pairing. It turns out that some transfer RNAs can read up to 3 triplets. This is particularly true where the 5' base on the anticodon is the non-standard inosine molecule. Here's a specific example of wobble with 3 alanine codons. The alanyl tRNA, which carries the anticodon 3' CGI, it has inosine as its first base. That anticodon recognizes the codons 5' GCU, 5' GCC, and 5' GCA. The mechanism of peptidal transfer from aminoacyl tRNA to the growing peptide chain involves the nucleophilic attack of an amino group from the aminoacyl tRNA in the A site on the carbonyl carbon of the aminoacyl tRNA in the P site. A new peptide bond is forged, and the link between the peptide and the P site and its tRNA is severed. During protein synthesis, the codons are read from the 5' to the 3' end of the messenger RNA. The protein is synthesized starting at the amino terminal end, proceeding towards the carboxyl terminal end. In prokaryotes, the first amino acid at the amino terminal end is usually F-met. Protein synthesis is terminated when one of the stop codons, UAG, UAA, or UGA, is encountered in the messenger RNA sequence. There are releasing or termination proteins which recognize these specific stop codons. The termination factors hydrolyze the bond between the protein and the tRNA. After hydrolysis severs the link to the tRNA, the protein diffuses away, and the ribosomal complex dissociates from the messenger RNA. Quite often, in prokaryotic organisms, several ribosomes can be translating the same messenger RNA simultaneously. With a ribosome near the 5' end of the messenger RNA, the nascent peptide would be quite short. If the ribosomal assembly is further down the message, the protein products would be longer. Near the 3' end of the messenger RNA, the protein product would be the longest. This type of assembly with multiple ribosomes carrying out protein synthesis on a single messenger RNA is referred to as a polyribosome or polysome. Once in a while, it's nice to have a picture that confirms the biochemical models we're learning about. Here's an actual bona fide electron micrograph of a polysome in action. Eukaryotic 80S ribosomes consist of a 40S small subunit and a 60S large subunit. The initiating amino acid in eukaryotes is methionine, not N-formyl methionine. The initiation of protein synthesis usually occurs at the AUG closest to the 5' end of the messenger RNA. The 40S ribosome finds this site by binding to the 5' cap and then scanning the RNA until AUG is reached. The regulation of translation in eukaryotes provides a means for regulating gene expression. There are a variety of things that can happen to DNA to change the sequence of bases. Genetic alterations in DNA structure are referred to as mutations. We can distinguish two common classes of mutations. A substitution or point mutation occurs when one base is substituted for another in the DNA sequence. This may affect the codon for one single amino acid, but it won't change the codons for any of the other amino acids because the reading frame hasn't shifted. A frameshift mutation occurs when a base is inserted or deleted from a sequence of DNA. This often causes profound changes in the amino acid sequence of the protein product because the reading frame is shifted. Frameshift mutations also commonly result in the formation of a shortened protein molecule. Here are two examples of what can happen when a substitution mutation occurs in DNA. The upper panel shows the normal DNA in black, its messenger RNA in blue, and the corresponding amino acid sequence, again in black. The middle panel shows what happens when a substitution mutation causes the T in the GCT alanine codon to be replaced with a G. The result is that the codon becomes GCG in the RNA. However, that triplet still codes for alanine, so there's no change in the protein sequence. This is called a silent mutation because the DNA and RNA are changed, but the protein isn't altered. The lower panel shows what happens when a substitution mutation causes the G in the GCT alanine codon to be replaced with a C. In this case, the DNA ends up with a sequence CCT, and the mRNA sequence is CCU. The CCU codes for proline, so there's a change in one amino acid in the protein sequence. That type of change in one amino acid is called a missense mutation. Some missense mutations have little effect on the function of the protein, while others cause profound changes. This slide gives two examples of frameshift mutations. Whereas substitution mutations are variable in their effects, frameshift mutations generally result in profound negative changes in the character and function of the protein product. We'll use the same DNA sequence to illustrate the effects of frameshift mutations. Again, the upper panel shows the normal DNA in black and its messenger RNA in blue, and the corresponding amino acid sequence, again shown in black. The middle panel shows what happens when an insertion mutation occurs in the TTT sequence, which codes for phenylalanine. When an A is inserted between the first and second T, that TTT codon becomes a TAT in the DNA coding strand. That's transcribed into UAU in the messenger RNA, which is read as tyrosine in the protein product. Downstream from that frameshift, the AGT, which codes for serine, becomes TAG. The TAG in the DNA becomes UAG in the messenger RNA, which is a stop sequence. Instead of a long protein, the gene now codes for a tetrapeptide. The protein is severely truncated and obviously non-functional. The lower panel shows what can happen when a deletion mutation occurs. In this case, one of the T's in the TTT sequence is deleted. The TTT becomes a TTA, changing the RNA to UUA, which replaces a phenylalanine in the peptide with a leucine. The frameshift also changes all the subsequent amino acids in the peptide. Although not visible in this part of the sequence, the most likely outcome is that somewhere fairly near to the deletion, the frameshift will produce a stop codon in the messenger RNA, and the protein will not only be a gibberish sequence of amino acids, but also will be severely truncated. Substitution mutations are often benign, but that's not always the case. The first human genetic disease to be characterized at the molecular level was sickle cell anemia. This disease is caused by a substitution mutation. The mutation results in an amino acid change at position 6 in the beta chain of hemoglobin. That amino acid is normally glutamate. The mutation changes a CTT sequence in the DNA to a CAT. That alters the encoded amino acid from glutamate to valine. The substitution of a neutral amino acid for an acidic amino acid causes a dramatic change in the solubility of hemoglobin and results in a severe, life-shortening disease. The normal red blood cell shown on the left looks like a biconcave disc. That's like a donut lacking a complete hole in the middle. In sickle cell anemia, the hemoglobin forms long, rod-like polymers that distort the shape of the cell, giving the sickled shape appearance that we see in the red blood cell on the right. Ultimately, these distorted cells burst and the hemoglobin is degraded. That causes an anemia, which is a reduced capacity for transporting oxygen in the blood. So, this is the grand finale, the coup de grace, the classic swan dive. You're now done with the lectures. Finished. No mas. Oh my goodness. The following photographs were selected at random. There's no theme, so be surprised. Many of the deer in the Olympic National Park are so tame you could feed them by hand. But don't try it or the park rangers will fine you. This mama and her fawns were running around the parking lot at Obstruction Point. My next-door neighbor has a dozen llamas. They are very photogenic, but they're otherwise totally useless. I'm sure that in Peru or Chile they serve some useful function, but not here. If you want a good big pet, get yourself a horse. My grandson Justice likes to ski. He wears these very cool reflective goggles. This is also a self-portrait of me. Again, note the very graceful way I hold the camera. I think this is the Washington State Ferry that runs from Bainbridge Island to Seattle. The light had a very lovely salmon-colored hue that evening. This view of downtown was taken from South Minneapolis. The rigid geometry of the downtown buildings forms an interesting contrast to the very relaxed